Welcome friends to a new session on genetics. In this session, we will discuss genetic recombination in bacteria such as conjugation, transduction and transformation. Genetic recombination in bacteria. First, let us see about microbial genetics. The progress in modern genetics is mainly by the experiments done in microorganisms like bacteria and virus. In these organisms, the DNA is not enclosed inside a membrane bound nucleus, but instead resides inside the cytoplasm. They have only haploid number of chromosomes. The chromosomes of bacteria, like those of other prokaryotes, are not enclosed in nuclear membranes. Hence, each character is controlled by only one allele. Bacteria are aptly described as the workhouse of the modern genetic world. You can see a detailed figure of the bacteria over here. Escherichia coli is the most commonly used bacterium for genetical studies living in the colon of man. It is microscopic, unicellular and rod shaped. It is covered by two layers, namely an outer cell wall and an inner plasma membrane. Many flagella arise from the body. The cytoplasm is colloidal in nature and contains granules of glycogen, proteins and lipids. Endoplasmic reticulum, Golgi complex, lysosomes, mitochondria etc. are absent. Ribosomes are present. Genetic information is carried by a single circular DNA present in the cytoplasm. This is called bacterial chromosome and it represents the nucleoid. In addition to bacterial chromosome, the cytoplasm contains plasmid, episome, fertility factor, sex factor. Plasmids are an extra chromosomal hereditary material that exists in an autonomous state. It contains some genes. A bacterium containing plasmid is called F plus and that lacks is called F minus. F plus cells have filamentous projections called pili. A single one is called pilus. During sexual process, a F plus cell can donate a copy of plasmid to F minus cell. Thus, the F minus cell can be converted into a F plus cell. Sometimes the plasmid can get integrated with the bacterial chromosome. Such cells are called HFR cells, meaning high frequency cells. HFR strains will, upon conjugation with an F minus cell, attempt to transfer their entire DNA. HFR strains vary in the chromosomal location of F factor integration and in the orientation of F minus factor, which determines whether the direction of gene transfer is clockwise or counterclockwise around the circular chromosome. HFR conjugation is the most natural mechanism of gene transfer in bacteria. The genetic information of bacteria is stored in a single main chromosome carrying a few thousands of genes and from 0 to several mini chromosomes called episomes or plasmids. Plasmids are highly variable in size, ranging from those carrying no more than 3 genes to those large enough to carry several thousand genes. Certain bacterial cells are known to harbor different types of plasmids in addition to the main chromosome. Some of them are fertility or F plasmids, which contain TRA genes capable of conjugation and result in the expression of sex pili. Resistance or R plasmids, which contain genes that provides resistance against antibiotics or poisons. Col plasmids, which contain genes that code for bacteriocins, proteins that can kill other bacteria. Degradative plasmids, which enable the digestion of unusual substances, example toluene and salicylic acid. virulence plasmids which turn the bacterium into a pathogen. Each bacterial chromosome is characterized today it contains a single circular molecule of DNA that exists in a highly condensed, that is folded or negatively supercoiled conformation in vivo. Bacteria have no sexual reproduction in the sense that eukaryotes do, because they have no alternation of haploid and diploid generations, no gametes and no meiosis. But the essence of sex is genetic recombination. Recombination is the rearrangement and reshuffling of genes resulting in new genotypes. Recombination, however, is undoubtedly important in the evolution of bacteria, just as in the evolution of eukaryotes. Bacterial recombination is brought about by the transfer of genetic material from one bacteria to the other. The bacterium which donates the genetic material is called donor and receives is called recipient. Three different processes have evolved that mediate transfer of genetic material from one bacterium to another, making possible the subsequent recombination events. They are: 1. Conjugation is the process which DNA from a donor is transferred to a recipient through a specialized sex pilus or conjugation tube. 2. Transformation involves the introduction of genetic material into a bacterial cell, and the third one, transduction, occurs when bacterial genes are carried from a donor cell to a recipient by a bacteriophage. The most obvious difference between these three processes is the mode of transfer of DNA from one cell to another. The three modes of recombination in bacteria can be distinguished by two simple criteria: sensitivity to the presence of DNase, that is deoxyribonuclease, and dependence on cell contact. These two criteria can be easily tested experimentally. The first criterion can be tested by simply adding DNase to the medium containing the bacteria involved in recombination. If recombination occurs in the absence of DNase but not in the presence of DNase, then the DNase sensitive recombination process, transformation, occurs. The protein coat of bacteriophage vector and the cell wall and the membrane enclosing the conjugation tube protect the donor DNA from degradation by DNase in the medium during transfer to the recipient cell during transduction and conjugation respectively. In this table, you can see the three different recombination events in bacteria. In the first one, transformation, no cell contact is required, it is sensitive to DNase. In the second one, transduction, no cell contact is required and it is not sensitive to DNase. In the third one, conjugation, the cell contact is required and it is not sensitive to DNase. Whether cell contact is required is tested by carrying out a so-called U-tube experiment, in which bacteria of two different genotypes are placed in opposite arms of a U-shaped culture tube. The two cultures of bacteria are separated by a semi-permeable membrane filter containing pores large enough to allow DNA molecules and viruses, but not cells, to pass through it. Conjugation cannot occur in the presence of DNase, and in the absence of cell contact it must be due to transduction. Now let us see about conjugation. It was first discovered in 1946 by Joshua Lederberg and Edward Laurie Totham. During conjugation, DNA is transferred from a donor cell to a recipient cell through a specialized intercellular connection or conjugation tube that forms between them. The transfer of genetic material is thus a one-way transfer during conjugation, just as in transformation and transduction. Cells that have the capacity to serve as donors during conjugation are differentiated by the presence of cell surface appendages called FPLE. The synthesis of F. pili is controlled by several, approximately 9 genes that are carried by a small circular molecule of DNA or mini chromosome, which contains about 94,500 nucleotide pairs, and it is called an F factor. The F factor can exist in two different states: the autonomous state, in which it replicates independently of the host chromosome, and the integrated state, in which it is covalently inserted into the host chromosome and replicates along with the host chromosome. Cells carrying an F- factor, that is donor cells, form conjugation tubes and initiate DNA transfer after making contact with the cells not carrying an F factor called F- cells, that is the recipient cells. A donor cell containing the F factor in the autonomous state is called an F plus cell. When an F plus donor cell conjugates with an F minus recipient cell, only the autonomous F minus factor is transferred. Both X conjugates, that is the cells that have involved in conjugation process, become F plus because the F factor replicates during transfer. Thus, mixing a population of F minus cells results in virtually all the cells in the new population becoming F plus, and this you can clearly see in this figure. The F factor can integrate into the host chromosome at any one of the many sites by a mechanism that appears analogous to the integration of the chromosome of a specialized transducing phage, namely a site specific recombination event. The integration of the F factor is believed to be mediated by short DNA sequences called IS elements, that is insertion sequence. Conjugation also occurs between HFR cells and F minus cells. In HFR cells, the plasmid remains integrated with the bacterial chromosome. During conjugation, the HFR cells transfers the plasmid or the plasmid with a portion of a bacterial chromosome. The F minus cells receives not only the genes present in the plasmid, but also a set of chromosomal genes as seen in this figure. Transfer is believed to be initiated by an endonucleolytic nick on one strand and at a specific site, that is the origin of transfer on the F factor. The 5' end of the nicked strand is then transferred through the conjugation tube into the recipient cell. The transfer is believed to be coupled to rolling circle replication. Now, let us see about transformation. Transformation was first discovered in pathogenic strains of Streptococcus pneumoniae, also called pneumococcus, in 1928 by Frederick Griffith. In this method, the donor bacterium liberates the DNA by the dissolution of the cell, and the DNA is absorbed from the medium by the recipient bacterium. Hence, the recipient bacterium is transformed into another strain. This is called transformation. There are two types of pneumococcus bacteria, namely virulent and non-virulent. Virulent strains have smooth carbohydrates capsules and give smooth colonies. Non-virulent strains have no capsules and give rough colonies. These two strains also differ in their antigenic properties and virulence for the disease pneumonia. Virulence is determined by genetic factors. When virulent strains are injected into mice, they kill them with pneumococcal injection. When mice are inoculated with non-virulent bacteria, the mice are alive. When mice are injected with virulent heat-killed bacteria, and the mice died due to pneumococcal infection. The analysis of dead mice shows that it contains virulent bacteria. The heat-killed virulent bacteria are responsible for the transformation of virulent bacteria into virulent smooth bacteria. Something from the heat-killed virulent bacteria was apparently transferred to the live non-virulent bacteria. This phenomenon is known as Griffith effect or bacterial transformation. It should be emphasized that although Griffith's experiment demonstrated the occurrence of transformation in pneumococcus and thus set the stage for the work of Oswald Theodore Avery, Colin Munro MacLeod and McLean McCarthy, they provided no evidence that DNA was involved in any way. The process of transformation can be divided into several stages. 1. Reversible binding of double-stranded DNA molecules to receptor sites on the cell surface. 2. Irreversible uptake of the donor DNA, at which time the donor DNA becomes resistant to DNase in the medium. 3. Conversion of the double-stranded DNA molecules into single-stranded molecules by nucleolytic degradation of one strand. 4. Integration, that is, covalent insertion of all or part of the single strand of donor DNA into the chromosome of the recipient. And 5. The segregation and phenotypic expression of the integrated donor genes or genes in the transformed or recombinant cell. Steps 2 and 3 may well be coincident effects of a single process. One attractive model for which there is supporting evidence in the case of pneumococcus proposes that a specific exonuclease or DNA translocase pulls one strand of donor DNA into the cell using energy derived from the degradation of the complementary strand. The first 3 steps in transformation, that is binding, uptake and degradation of one strand of the double-stranded DNA, are not specific for homologous DNA. In fact, competent bacteria will carry out these 3 processes equally well given calf thymus DNA or other foreign DNAs. However, in integration or DNA recombination step is specific for homologous DNA. This is not to say that the integration of segments of heterologous, that is foreign DNA, never occurs. If it does occur, however, it does so at frequencies very much lower than the frequencies observed using homologous DNA. Although very small amount of DNA, about 500 nucleotide pairs, are taken up by the competent cells required for integration to occur. E. coli cells are more likely to incorporate foreign DNA if their cell walls are altered so that DNA can pass through more easily. Such cells are said to be competent. During integration, a single strand of donor DNA is physically inserted into the recipient chromosome, replacing a segment of one strand of the recipient chromosome. In most transformation experiments, donor DNA fragments are about 20,000 nucleotide pairs or about 1/200 of the total chromosome in length. Alpha complementation is a method for bacterial screening that have been transformed with a plasmid vector carrying the N-terminal coding sequence for beta galactosidase of the lactoferrin. The LAC-Z gene is required for galactosidase metabolism. The PUC plasmid carries the alpha fragment of the LAC-Z gene, which is the amino terminus of the protein and is non-functional alone. The omega fragment is found in E. coli chromosome, which is the carboxy terminus of the protein and is non-functional alone. When the alpha and omega fragments interact, they become functional and beta galactosidase protein can be produced. This interaction is known as alpha complementation. It is useful during the screening of beta galactosidase assay. Now, let us see about transduction. It was discovered by Norton David Zinder and Joshua Lederberg in 1952, and it occurs when a bacteriophage particle carries a segment of the chromosome from one bacterium to another bacterium, facilitating subsequent recombination of the genetic markers of the two cells. Two different types of transduction are known: generalized and specialized transduction. First, let us see about generalized transduction. In generalized transduction illustrated in this figure, a random or nearly random segment of bacterial DNA is wrapped up during phage maturation in place of or along with the phage chromosome in a few progeny particles called transducing particles. Generalized transducing phages can therefore transport any gene of the donor cell to the recipient cell. Since all the genes of the donor are represented in a population of these transducing particles, this type of transduction was named generalized transduction. In some cases, generalized transducing particles contain only bacterial DNA. In other cases, they contain both phage and bacterial DNA. Initially, in this figure, you can see the phage infects bacterial cell. Later, host DNA is hydrolyzed into pieces and phage DNA and proteins are made. Further, a bacterial DNA fragment is packaged in a phage capsid. In the next step, transducing phages infect new host cells where recombination can occur. And finally, the recombinant have genotype A plus B minus different from either donor, that is A plus B plus, or the recipient A minus B minus. The structural aspects of a bacteriophage includes one head which stores genetic material, either DNA or RNA, may be single or double stranded, may be linear or a closed loop. 2. Sheath, which provides a path for genetic material to be injected into the host cell, and the third one is tail, which attaches to the host cell. These three structures are clearly seen in this figure. Bacteriophages have been classified into two types on the basis of their interaction with the bacterial cell. They are virulent phages, always multiply and lyse the host cell after infection, and temperate phages have a choice between two life cycles after infection. One, they can either enter the lytic cycle, during which they reproduce and lyse their host cells just like virulent phages, or alternatively. The different stages of lytic cycle are as shown in this figure. 2. They can enter the lysogenic pathway, during which their chromosomes are integrated into the chromosomes of host and replicate like any other segments of the host chromosomes. The different stages of lysogenic cycle are as shown in this figure. Generalized transduction is mediated by some virulent bacteriophages whose chromosomes are not integrated at specific attachment sites on the host chromosome. Generalized transducing particles are produced during the lytic cycles of these phages. In the first stage of the lytic cycle, the phage attaches to host cell and injects DNA. In the second stage, phage DNA circularizes and enters lytic cycle or lysogenic cycle. In the third stage, new phage DNA and proteins are synthesized and assembled into virions. And in the fourth stage, cell lyses and releases phage virions. Now, coming to the lysogenic cycle, the phage DNA circularizes and enters into the lysogenic cycle, and in the next step, phage DNA integrates within the bacterial chromosome by recombination, becoming a prophage. In the next stage, the lysogenic bacterium reproduces normally, and finally, the prophage may excise from the bacterial chromosome by another recombination event initiating a lytic cycle. Not all virulent phages mediate transduction. The T-even bacteriophages, that is T2, T4 and T6, for example, degrade the host DNA and neutralize the mononucleotides produced for the synthesis of phage DNA. Of the generalized transducing phages, E. coli phage P1, Salmonella phage P22 and Bacillus subtilis phages PBS1 and SP10 have been extensively used for genetic fine structure mapping. After a transducing phage injects a fragment of host DNA into a recipient cell, that DNA may either be integrated into the host chromosome in a manner similar to the integration to the transforming DNA, except that the integrated segment is double stranded or remain free in the cytoplasm. If it is not integrated, it will not be replicated and will be transformed to only one progeny cell during cell division. The genes located on the transducing chromosome fragments may be expressed even if they are not integrated. Cells carrying non-integrated transducing fragments are called abortive transductants, and the process is called abortive transduction. Abortive transduction is a transduction event in which the fragment of donor gene does not incorporate in the recipient chromosome, and because the DNA fragment lacks an origin of replication, only inherited by one daughter cell at each cell division. Now, let us come to the case of specialized transduction. In this figure, you can see this process is mediated by temperate bacteriophages whose chromosomes are able to integrate at one or a few specialized attachment sites on the host chromosome. The chromosomes of temperate phages of this type are thus capable of both 1. Autonomous replication, that is, replication independent of the replication of the host chromosome, and 2. integrated replication, that is, replication as a segment of the host chromosome. The example for this is episomes. The different stages of specialized transduction are as follows. In the first stage of specialized transduction, bacterial cell has prophage integrated between genes A and B. In the next stage, prophage DNA exits incorrectly, taking adjoining bacterial DNA with it. Now, coming to the next stage, phage particles carry bacterial DNA along with the phage DNA. Later, transducing phages infect new host cells where recombination can occur, and further the recombinant have genotype different from either the donor or the recipient. Integration of the chromosome of a specialized transducing phage such as the coliphage lambda involves a recombination event between the circular intracellular form of the phage chromosome and the circular bacterial chromosome at specific sites on the two chromosomes. This site specific recombination event results in the covalent linear insertion of the phage chromosome into the chromosome of the bacterium. In its integrated state, the phage chromosome is called a prophage. The lytic gene of the virus, those involved in viral reproduction and lysis of the host, are repressed when the chromosome is in the prophage state. A bacterium harboring a prophage is called lysogeny. The prophage-host relationship is called lysogeny. Temperate phages undergo rare spontaneous transitions from the lysogenic or prophage state to the lytic state. Such transitions can also be induced, for example, by irradiation with ultraviolet light. During the switch from the lysogenic state to lytic growth, the prophage is excised from the host chromosome and commences replicating autonomously. The excision process is site specific, like the integration process. The site specific integration and excision process are catalyzed by enzymes encoded by phage genes. The excision process is usually very precise in cutting out the phage chromosome in exactly the form in which it is excised prior to its integration. Occasionally, however, the excision event occurs at a site other than the original attachment site. When this happens, a portion of the phage chromosome and a portion of the bacterial chromosome is excised with the phage DNA. Such mistakes during prophage excision are responsible for the formation of specialized transducing particles. Now, let us summarize the following discussions on conjugation, transduction and transformation. The progress in modern genetics is mainly by the experiments done in microorganisms like bacteria and virus. Bacteria are aptly described as the workhouse of the modern genetic world. The most commonly used bacterium for genetical studies is Escherichia coli, living in the colon of man. It is microscopic, unicellular, rod shaped. Genetic information is carried by a single circular DNA present in the cytoplasm. The cytoplasm contains another circular DNA called plasmid. Bacteria have no sexual reproduction, but the essence of sex is genetic recombination. Recombination is the rearrangement and reshuffling of genes resulting in new genotypes. Three different processes have evolved that mediate transfer of genetic material from one bacterium to another, making possible the subsequent recombination events. They are: 1. Conjugation, which is the process in which DNA from a donor is transferred to a recipient through a specialized sex pilus or conjugation tube. 2. Transformation, which involves the introduction of genetic material into a bacterial cell. And 3. Transduction, which occurs when a bacterial's genes are carried from a donor cell to a recipient by a bacteriophage. Now, I will give you certain assignments based on the discussions what we had. 1. Define genetic recombination. 2. List 3 types of bacterial genetic recombination seen in nature. 3. Briefly describe the mechanisms for transformation in bacterium. 4. Explain Griffith's experiment. 5. List out the criteria that can be distinguished in the 3 modes of recombination in bacteria. Now, I am giving you certain references for your further reading. The first one is a book entitled Cell and Molecular Biology, authored by D. Robertis and Robertis in 1991, published by Leah and Febiger, Washington. Second one is with the title Genetics and the Origin of Species, authored by Dobzhansky in 1961, published from Columbia University Press, New York. The third book is with the title Principles of Genetics, authored by Gardner and Nustad in 1984, published by John Wiley, New York. The fourth one is with the title Genes, authored by Levin 1994, published by Oxford University Press, London. The fifth book is with the title Polyploidy, authored by Lewis in 1980, published from Plenum Press, New York. A sixth book is with the title Chromosome Technique, Theory and Practice, authored by Sharma and Sharma in 1980, published by Aditya Books, New Delhi. A seventh reference is with the title Principles of Genetics, authored by Sinnott, Dunn and Dobzhansky in 1958, published by McGraw-Hill, New York. I am also giving you certain websites for your further reference. The first one, www.genetics.org. Second one, www.theagricos.com. We have come to the end of this session. We will meet in another session with a new topic. Till then, bye.